Hi everyone, here goes a very quick introduction to Fisher's test of significance. I will restrict myself to just a discussion of the workings of the test in this presentation. I will leave for another presentations further comments if necessary. Tests of significance are very easy, very simple to do, and actually very convenient. 5 steps is all what you need to do a test, and not only that, it's also a test that you don't need to necessarily think a priori before you do the research data. You could as well collect your research, collect your data, and then apply the test once you are ready to interpret those results. So let's start with this step as step 1. Let's say you have two groups. You have the experimental group, to which you have given treatment, and you have your control group. Later on, you pass a common task to measure performance, for example, and you find a difference between the two groups. You use the statistics that are necessary to measure that difference. In this case, would be a t-test for independent groups, which will give you a standardized score of the difference between those two groups. In our case, a score of 2.25. Right, so you got your research results, you got the standardized t-value. What does it mean? Here is where the test starts. So in order to be able to interpret that test score, that difference between groups, in a sensible manner, you need something to compare with, a standard against which you can compare that research result. And this is basically a distribution to calculate the probability of the data. Okay. Now this distribution needs to be an exact distribution. So it cannot be based on the actual research data, because that you don't know what you're going to get, therefore you cannot establish that distribution. But you can do that, for example, with 0. 0 means that there is no difference between the groups, or in another occasion there are no correlations between variables. So 0 is very exact, it's 0. That's basically your starting point. And that's why it's called the null hypothesis, because you expect no difference between groups or no correlation between variables, etc. So this distribution, the null distribution, is actually the distribution of scores that happens when you know that there is no difference between two groups or no correlation between two variables. Alright? So the null distribution actually represents the distribution of data when the true difference between groups is 0 in our example. And this distribution is a particular distribution. It has been generated from information, partly from information from your sample, and also from other theoretical information. And from your sample it gets information such as the sample size will be transformed into the degrees of freedom and standard deviations in order to standardize the distribution. Alright. So in our case, the distribution that we will be using for constructing our research data will be a t-distribution with 30 degrees of freedom. Now, one interesting thing is all potential values that we may get from research are able to be located within that distribution, either very close to 0 if the mean difference is not much, or further into the tails. Okay, but all potential t-values will be located under the null hypothesis. Not only that, but because this is a fixed distribution, we know its shape, we know its probability density, all those potential t-values will also have an associated probability. So for example, in this distribution that we have here, a t-value of 1 will always have a probability of occurrence under the null distribution of 16%. That is about under this particular distribution with 30 degrees of freedom. Alright. Once we have the distribution, then the only thing that we need to do is to locate our research data within that distribution. And this is pretty much what the test does. So our original research data of 2.25 t-scores is located at that particular point in the distribution of the null hypothesis. What is perhaps more interesting to us is to know its associated probability, which is 1.6%, one tail in this case. So not only the location, we get information not only about the location in the distribution, but also about the probability of the data under that location. Now, these two pieces of information conveys the same information to us. Okay, so you could use the t-value, or you can use the probability, although perhaps the p-value is the one that is more useful for purpose of the test. The main difference between the two is that the t-value is at precise location on the distribution, while the p-value is an accumulated probability. Okay, so it's includes not only the probability of that particular t-value, but also of any other more extreme t-value. But this is just a note of interest to you. Our first step will be to assess the statistical significance of the results. Alright? Basically, to assess how low the probability of the result is on the null distribution. Now, you can't assess this significance by just having a subjective assessment of if that p-value is low. But if you don't know much, you could also use conventional levels of significance. Typically, we use conventional levels of significance such as 5% to represent, the starting level of significance, or 1% to represent high significance, and 1 per mil to represent extremely high significance. Right? Now, what is important here is that these conventions are approximate rather than exact. So, for example, in our case, with a p-value of 1.6%, this is quite close enough to the conventional level of 1%, so we could interpret it as being highly significant. Okay? So, basically, a low p-value is what we use to assess significance. And the p-value here has the role of being evidence against the null hypothesis. So the lower the p-value, the more extreme the result, then the stronger the evidence. So obviously, if you get a significance closer to 1%, is stronger evidence, but even much stronger if you get a p-value closer to 1 per mil, or even much more extreme. And finally, you need to interpret the results. And now here is the tricky part. Or is it not tricky, but something to take into account. A test of significance always offers a duality that you cannot escape. That is, either you got a extreme result under the null hypothesis, but still the null hypothesis explains the difference between the groups. That is, the probability could be lower, but it was just a rare event that happened to you, or else the null hypothesis may not explain the results satisfactorily. Now, this is a leap of faith. This is a decision, an assessment, or an interpretation that you need to make. The test itself cannot help you resolve this interpretation. It doesn't provide you with the answer. So this is a conclusion that you have to reach on your own. Alright, and pretty much this is all what it has to do with the test. Nothing more than that. Thanks for watching, and if you want more information, here some references, especially the one from Khan Academy is very good for those who are learning to do hypothesis testing by hand. And with this, we finish our brief introduction to Fisher's test of significance.